What could be more exciting than unfolding the code of life? DNA holds information about our past as well as our predispositions. But what is this molecule made of? DNA is composed of a long chain of nucleotides, which consist of a nitrogenous base, a pentose sugar, and a phosphate group. What makes nucleotides different from each other is their nitrogenous base. Nitrogenous bases fall into two categories. The first category is purines, like adenine and guanine, represented by the letters A and G for short. Purines are composed of a double ring structure, one 5-membered ring attached to a 6-membered one. The other category of bases are known as pyrimidines, like cytosine and thymine. Just like adenine and guanine, these can be called by their abbreviations of C and T. In the case of RNA, thymine is replaced by another pyrimidine known as uracil, which is represented by, you guessed it, the letter U. Unlike purines, pyrimidines are composed of a single 6-ring structure. In DNA, the pentose sugar is deoxyribose, and in RNA, it's ribose sugar. In 1953, the discovery of the structure of DNA paved the way to further understand our genetic makeup. Attempts were made to sequence this code, but efforts were long and costly. It wasn't until 1977 that a relatively fast and cheap sequencing technique was developed by Fred Sanger. This new technique revolutionized the way DNA sequencing was read, expediting the process. It is known as the dideoxy chain termination method, which, as the name suggests, relies on dideoxynucleotides for chain termination. In this method, DNA is first heated to high temperatures to separate it into two strands through a process called denaturation. The DNA is then placed in 4 different tubes with a primer, polymerase, and nucleoside triphosphates. In each tube, a limited quantity of a single type of the 4 dideoxynucleoside triphosphates is added. Each is labeled with a different fluorescent dye. The resulting fragments of DNA are then separated using gel electrophoresis and a laser scanner to read the fluorescent labels of each fragment. The fragments can be separated by size from largest all the way down to a single nucleotide. These fragments are then ordered so that the sequence can be read by the colors of the markers. These indicate the position and type of nucleotide within the sequence. Now that we understand the structure of DNA down to its base and sequencing, doesn't it seem simple? Hardly. The code of life doesn't end here, but innovations like understanding DNA structure sequencing have paved the way to great scientific breakthroughs. Now, that's a code worth cracking.